Welcome everyone to this week's edition of the Extracellular Vesicle Club. So before we get started today, I have a couple of announcements. The first one is that next week we will not have a regular Wednesday session, and the following week we will not have a regular Wednesday session, and that is because of the ISEV annual meeting. So the ISEV annual meeting this year is virtual. It will start with an education day next Wednesday, the 12th, and then it will continue with some satellite events throughout the intervening week. And then finally, the next week, we will have the main ISEB meeting. So there's still time to register for the meeting. It's a lower price this year because we're not in person. We do have to pay for the platform that we're using, but it's a pretty good deal to attend the world's leading meeting on extracellular vesicles. So please attend if you can. And the other announcement is that since we are not having the meeting for 2 weeks, we're actually going to have a special session next Monday. So it will be a Monday session at the regular time, but we will be sending out some notifications about that soon. All right, well, today we have a collaboration of ISEV and the Student Network on EVs. And Camille Trinidad is here representing the Student Network, and she's going to be moderating. So I will hand it over to Camille to introduce our presenter today and also our special guests. Camille, go ahead.
Hi, thanks, Ken, for that. Hi, everyone. Our presenter for today is Nishtha Bhargava. She obtained her master's in science degree in biotechnology from Punjab Technical University. She's currently a PhD student at the Academy of Scientific and Innovative Research in the Institute of Genomics and Integrative Biology in India. Her studies are focused on how small EV cargo changes when cells are under different stresses using mammalian culture. So Nishtha is presenting this paper, which was published in PLOS Biology last month. And today we're also fortunate enough to have Stefano Pluchino and Luca Perugiotti Giametti. And their lab is found in the University of Cambridge, Department of Clinical Neurosciences. And they'll be here to help us answer questions during the discussion at the end of this presentation. And so thank you for joining us today. If you have anything to say before Nishtha is going to present your paper, you're welcome to say something about the paper that we're talking about today.
I think I can proceed. So basically, this is a follow-up of other papers that were published in the lab, where we focused on specifically neural stem cells and how they're able to secrete EVs to modulate and target neighboring cells. We did publish several papers in that area, but the things that we were mostly interested in were also to understand if there's any possibility of intercellular communication. They might be linked to mitochondrial transfer. And the idea came after this paper from Englo was published in Nature a few years ago, showing transfer of mitochondria in the central nervous system from astrocytes to neurons. And then we did some studies to understand if this was something that was related only to the CNS, and we found actually that it was not. So it seemed to be something that it was quite diffuse inside the biology and the realm of science, but no one has ever looked into neural stem cells. So that's why we went into this journey of looking specifically whether this could happen in our cells and specifically in EVs.
Great. So let's go ahead and move to the paper. So Nishtha, we see your screen really well, and take it away. Thank you.
Thank you. Yeah. So this paper I saw has been published recently in PLOS Biology, and so has already given the introduction to it. What I was interested when I looked at this title, what I was interested to see was how can really mitochondria fit into vesicles? Because I work with small extracellular vesicles, and for me, the issue of size, things fitting into that tiny nanometer size vesicle is something that's always at the back of my mind. So I was interested in, first of all, seeing how they have proven that mitochondria are pretty much inside extracellular vesicles. But like Sir said, this is from the title, the protagonists of this story are basically neural stem cells, and that they traffic mitochondria through EVs, and the role of exosomes has also been explored. but what they transfer it to and in what context this is further investigated doesn't appear in the title, which is the mitochondria are transferred to macrophages, which is shown in the paper, in the second half of the paper, sort of. And they have told us that this whole process influences how inflammation occurs in the body and through macrophages. And they have used in vitro cell culture model for most of the work. And they have also done some in vivo study at the end to prove that the same things that we see happening inside the cells do in fact replicate in vivo as well. So this has come up after a lot of other studies which have happened in the past decade. So things like post-ischemic neuroprotection and what are the mechanisms, cellular mechanisms that help these processes. The fact that extracellular vesicles from neural stem cells can transfer molecules like interferon gamma to activate signaling pathways in receptor or target cells, and that they are independent metabolic units that have certain enzymatic activities. All these things clearly tell us that there is a possibility of extracellular vesicle carrying entities that can affect or modulate the metabolism inside the target cells. And specifically talking about mitochondria, mitochondria have been shown to be transferred through various mechanisms like tunneling nanotubes and also through EVs. And it is also known that in the literature very recently that the quality of mitochondria that is transferred sort of influences the inflammatory processes that are happening inside the target cells or in the tissues that are concerned. So going by this literature, the group has tried to sort of elaborate how mitochondria are transferred through extracellular vesicles and what effects they have in the target cells. So there's a lot of data and a lot of, so I'll straight off go into it. First of all, they have used untargeted multiplex TMT tag-based proteomics to inquire what all protein cargo is contained inside the EVs. By this process, they have basically put isobaric tags on different samples and then found what all proteins can be identified in neural stem cells, in the EVs that they generate, and also in the exosomes. By exosomes here, what they mean are CD63 enriched vesicles. And through this multiplex proteomics, they found close to 10,000 proteins in this whole set. So using these 10,000 proteins, they further do a gene ontology, cellular component annotation, as well as a relative abundance calculation, particularly in context of mitochondria later on. So with the gene ontology, cellular component annotations told them that the proteins indicating exosomal and mitochondrial proteins, mitochondrial localization, are enriched in the EVs compared to the neural stem cells, whereas as a negative control sort of, the nuclear localized proteins are depleted. Secondly, the proteins, they go further level down, deeper, by looking at whether the proteins that localize to the various subcompartments of mitochondria, such as the intermembrane space and the matrix, whether they are also enriched in this EV set. And indeed, they find that all the three subcompartments, proteins in all the three subcompartments, show an enrichment in EVs when compared to the neural stem cells that are producing them. Next, they further analyzed this and they found that both mitochondria encoded proteins of the mitochondria and nuclear encoded proteins of the mitochondria are enriched in EVs compared to the neural stem cells that are the originators of these EVs. They validate this with the immunoblotting. As you can see here, they have taken one counterpart from each of the complex 1, 2, 3, 4, and 5. 2, 3, 4, and 5 actually here. And you see that in the EVs, there is an enrichment of a lot of those components compared to the neural stem cells themselves. And as a positive control, they have used the mitochondrial isolates. So EVs are enriched in mitochondrial complex proteins. Here they have also looked for EV positive markers such as CD9, TSG101, and PDCP6IP. And Golga2 is used as an EV negative marker, and indeed it is not present in the EV isolates. Next, what they wanted to see was whether EVs contain any mitochondrial DNA, or basically looking at whether only the mitochondrial proteins are present in the EVs, or is it a possibility that the entire organelle or a part of the organelle could be present. So with respect to that, they found that, interestingly, the mitochondrial gene ND1, when it was amplified through PCR, the EVs did show up signal for that, which means mitochondrial ND1 is present in the EVs. But when they try to amplify a nuclear protein, which is nuclear gene, the protein produced by which is localized in the mitochondria, they could not find it in the EV or the mitocondria, which clearly indicates that there is possibility that intact mitochondria might be present within extracellular vesicles that are isolated through ultracentrifugation, as they have done this till here. Next, they want to see whether the various different isolation protocols have any bearing on this result. So to do that, they used isolation protocols that do not involve ultracentrifugation. So they used precipitation reagents from Qiagen and Invitrogen, and also some modified protocols from the past literature. And here also you can see that EVs that are isolated through these protocols are pretty much showing the same signals. They do show an enrichment of mitochondrial complex proteins and the membrane protein TOM20, as well as they show CD9 and other positive markers, and Golga2 is absent. So basically, precipitation-based isolation protocols yield Golga negative, but CD9 and mitochondrial protein enriched EVs, which is what they found with the ultracentrifugation protocol also. Next, they wanted to check if they enrich the CD63 fraction. So this they do by means of immunoaffinity purification. And they want to check if CD63 enriched vesicles, which purportedly we can call exosomes, would also show the mitochondrial protein enrichment. And indeed, you see here in this lane that the CD63 enriched EVs also show an enrichment of mitochondrial complex proteins. So again, lastly, what they did was to introduce an ultrafiltration step in the ultracentrifugation method of isolating EVs, which is present in this paper. And what they see is that the mitochondrial complex proteins significantly reduce, though they do not disappear, but they significantly reduce upon filtration during EV isolation. So basically, the processes that whatever isolation method is being used, the mitochondrial complex proteins are enriched in the vesicles all the time. So that is not a cause of bias in whatever the results are being produced. Next, they wanted to compare between proteins that are present in the crude EV preparations, that is the ones that are obtained through ultracentrifugation, and the CD63 enriched ones, which we're calling exosomes here. So there is a huge overlap between these two, between the protein cargo of these two vesicle preparations. Then, even then, there are several proteins that are selectively depleted in the exosomal fraction, or the CD63 positive enriched fraction. So those several proteins are selectively depleted in exosomes, as you can see in this graph. These blue dots, which show a negative log2 ratio, they are significantly depleted in the exosome preparations, although a few also show enrichment. So they wanted to see whether mitochondrial subcompartment localized proteins are enriched in these exosomes also. So just like they found in crude EV preps, they did find that exosomes are significantly enriched. So these red circles that you see that are on the right of the central margin of this log2 are enriched, and of all the three subcompartments, many proteins are found to be enriched in exosomes as well. as you know that isolation of exosomes, further purification of exosome isolates can be done through sucrose density gradient. And they did the same with those exosome preparations. And what they found was that even after the sucrose density gradient purification, they could find the mitochondrial complex proteins in the fractions that correspond to the density of exosomes. These fractions, they show all the complex proteins, as you can see here in 6, 7, and 8 also, and TOM20 as well, which is a membrane protein of the mitochondria. TSG101, which is a marker for small extracellular vesicles or exosomes, is present here, and GOLGA2 is absent. So sucrose density gradient of exosome preparations also shows mitochondrial complex proteins presence, and it also shows the presence of mitochondrial DNA. So the same gene, mitochondrial ND1, is amplified by PCR, and we can see that all these fractions show the presence of this DNA, but the nuclear DNA, SDHD, the protein of which is actually localized to the mitochondria, is completely absent from this preparation. So from this set of experiments, they conclude that NSE-derived exosomes, as well as crude EB preparations, harbor mitochondrial proteins and mitochondrial DNA, irrespective of the isolation protocol employed. At least for the ultracentrifugation method, ultracentrifugation with ultrafiltration, and for precipitation protocols for isolation of EVs, there is no bias towards any one in any protocol towards these results. They come up every time we do any EV isolation. Now, the next question that they want to answer is whether the mitochondria that are present in these EV preparations, are they intact or are they fragmented, or are there just proteins of mitochondria that are present in these EVs somehow? So for that, they first of all collected morphological evidence for presence of mitochondria in the EVs. So using TEM, they have looked at the size distribution of the particles, and they derive the proportion of EV preparation that shows mitochondria-like size and morphology. So these, all the dots are all the particles that have been checked in TEM, and the purple ones are the ones that that show mitochondria-like size and morphology. So about 27.81% or about 30% of the particles show mitochondria-like morphology, which means that there is presence of mitochondria-like particles in these preparations. Second approach that they took was nano-FCM analysis to derive the proportion of EVs that are positive for mitotracker red. So mitotracker red is a red that is specifically binds to mitochondria, and CD63 is the second signal that they have used to see whether EVs and mitochondria come up in the same quadrant of the nano-FCM analysis. So if you see, this is mitotracker red axis and the CD63. So something that is positive for both, which we can imagine to be vesicles that contain mitochondria, will fall in Q2 here. So 26% of this graph is Q2. Q2 and Q3 together combined are the particles that contain mitochondria. So that makes about 33.52% of the entire population that is checked in the nano-FCM. Of this 33.5%, more than 3/4 is positive for both CD63 as well as mitotracker, which means that about 3/4 of the particles that carry or are containing mitochondria are actually EVs. Notice here that the mitotracker is absent from these mitodepleted fractions. So these fractions are basically from the crude EV prep, where they have got the part which is mitochondria from the cell preparation after mitochondria depletion. They have got the mitodepleted fractions. So here you see that it is negative for mitotracker, and also very little of it falls in the first quadrant, which is CD63 positive. So next, what they do is the antibody-based detection of mitochondria presence in extracellular vesicles. So using cryo-TEM and gold nanoparticle immunolabeling, they have looked at the sort of co-localization of the two beads in a given site. So here you can see that the 10 nanometer gold nanoparticles that are that have affinity towards TOM20, which is the mitochondrial protein, mitochondrial membrane protein. It indicates mitochondria here. And in the inset, which is the expansion of this portion, you can see that there is a double membrane, which is visible here. So there seem to be certain free mitochondria with a double membrane. But then they also, they did a double immunolabeling, which is basically 10 nanometer gold particles for TOM20 and bigger 20 nanometer particles against CD63 of vesicles. So here you can see that some of them are free, whereas some of them, like these, they show that CD63 and TOM20 are present in sort of the same space. And from the, if you blow up this inset, you can see that there are 3 membranes visible here, which include sort of one layer of the EV and other 2 of the mitochondria that was also visible here. So from morphological analysis and gold immunolabeling, it appears that mitochondria are indeed present in EVs. And since we are looking at a matrix in a membrane protein here, most probably they are intact. The very dependable hallmark of an intact mitochondria is basically the electron transport chain and the activity, basically the mitochondrial activity. And to prove that point finally, they have done a lot of activity assays. For example, they have looked at the JC1 ratio. This is a dye that fluoresces either green or red depending on the polarity of the mitochondria. So CCCP is a compound that uncouples the electron transport chain and leads to its decline, basically, in the mitochondria. So if you look at the cells, the neural stem cells, there is an absolute decline in the red to green JC1 ratio when CCCP is present. So in mitochondrial preparation from these cells, you can see that the JC1 ratio declines with treatment of CCCP, and same way in the EVs. But in the mitochondria depleted fraction of the EVs, there is no change because there are no intact mitochondria in the first place to actually uncouple the ETC in. So you do not see an effect here. So from this, we can conclude that EVs in fact do have an intact mitochondria within them. Then they also looked at the oxygen consumption rate. So this is oxygen consumption rate over time. And they have used substrates of complex 1, complex 2, so and so, and also inhibitors like rotenone is an inhibitor of complex 1 to see how the oxygen consumption changes with time only with respect to the EV preparations. So if EV preparations are consuming oxygen and they do show a sudden decline when potassium cyanide, which is basically a poison for the mitochondrial respiration, is given to the EV preparation. That means that most probably intact mitochondria are present in the EVs. Similarly, they look at the oxygen consumption rate through another assay, and they do find this again only done on the EVs, and the consumption rate is high. So this also indicates intact mitochondria. Then they do a native page, a blue native page, so that the complexes are bound to each other and they don't fall apart. So that is why a native page. And they look at the presence here simply. So this is just a Western blot of EVs, mitochondrial preparations, and neural stem cell preparations and they have checked for all the 5 complexes. And you can see here that the complexes that the mitoprep is showing, EVs are also showing the same complexes. This hints towards the fact that intact mitochondria are present in EVs. And lastly, they have done an activity in gel activity assay on this setup, and you do find that the EVs are also showing activity in complex 1, 2, and 4, just like mitochondria, although not to the extent as mitochondria, but more than the cells. So there seems to be an enrichment of intact mitochondria within EVs. So all this set of data tells you that the mitochondria that are present in EVs are certainly intact. And from this, we can see that the mitochondria we get, the mitochondria depleted fraction of the EVs does not contain any intact mitochondria. So the process seems to be working, and intact mitochondria are present in EVs. So now that these intact mitochondria are present, next what anyone would want to ask is whether these EV-associated mitochondria can do something in cells. So mitochondria depleted cells are called Rho zero cells. So Rho zero cells, and this particular cell line, L929, does not. L929 Rho cells have been produced, and when you plate them, you're supposed to give them uridine in order to allow them to survive, because they cannot survive without uridine. They're oxotrophic for uridine. So when you withdraw uridine from them, they will start to die. And as to see the effect of EV-associated mitochondria, the EVs that have come from NSCs earlier. So basically, NSCs have been transduced to contain mito-DS red. So their mitochondria is stained, basically. So they prepare mitochondria from the neural stem cells, and they also prepare EV isolates from the same cells. Both of them will have some amount of red fluorescence because of the mito-DS that is labeling the mitochondria. So once uridine is withdrawn from these Rho zero cells, which do not have active mitochondria in them, then when you treat them with either mitochondria or the EVs, you look at the effect that they're having. So there are three things that they have checked, which is early immunofluorescence to check whether mitochondria are entering, and to see their effect on cell survival by counting the cells, and to see whether mitochondrial DNA is transferred. So using this model, first of all, they look at the immunofluorescence. So here you see mito-DS red is in red, DAPI is to stain the nucleus, and phalloidin is used to basically mark the cell boundary. And when it comes to only mitoprep from these neural stem cells, you see that they are not sort of entering into the cells. They are either on the surface or in the surrounding. But when it comes to EVs, in the same ratio, that is 1:30, you see that in the inside it is much clearer that they are either interacting with the membrane or entering the cells. This is after 6 hours of treatment, and after 24 hours of treatment, when they look for mito-DS red positive cells, like the target cells that are turned mito-DS red positive because of mitochondria transferred into them, you see that proportion is much higher for EV-associated mitochondria than for just the mitochondria preparation. This is after 24 hours of treatment. So first of all, mito-DS red positive EVs are taken up by L929 Rho zero cells as early as, 3 hours, not 6, 3 hours. And secondly, L929 Rho zero cells incorporate mitochondria from these EVs. And so your first first checkpoint is covered. Next, they check for the cell viability or growth in presence of these EVs. So as I said before, if you do not provide uridine to these Rho zero cells, they will not be able to survive. So when you remove uridine, you see a large amount of cell death, basically. And as you provide them with either mitochondrial preparations or EV preparations, you check whether the cell growth is rescued or not. Also, basically, whether the cells are able to survive or not. So in cells that do not have any mitochondria, if you give them mitochondria, they become independent of presence of uridine in the medium, and then they are able to survive and to further divide. So if you count the DAPI positive cells per unit area of the cell culture, you basically see that there is an increase when it comes to EV-associated mitochondria. So these two, whether you give them 1:30 ratio or 1:90, the increase is quite significant. So that means EV-associated mitochondria are supporting the growth or allowing the survival of Rho zero cells, which cannot survive without uridine by themselves in the media. Lastly, they looked at whether mitochondrial DNA is found in Rho zero cells. They're not supposed to have any mitochondria in the first place, but if they are taking up mitochondria through the media, then you will find mitochondrial DNA. So they do check this on the 16th day, and like previously, the same gene, mitochondrial ND gene, ND3 here, is checked for through Sanger sequencing. And Rho zero cells treated with EVs show this DNA, as well as Rho zero cells treated with mitochondria. So basically, EV-associated mitochondria are incorporated by and they restore the mitochondrial function of persistently mitochondria DNA depleted target cells. Next, since we now know that the mitochondria that EVs carry are intact, and these intact mitochondria can get into target cells, and they do give that advantage, whatever mitochondrial activity and mitochondrial function can give to a certain cell. Next, the investigators wanted to see if the known effect of known modulatory effects on neuroinflammation is also a sort of a avenue where this process is having any effect. So since they already have this report, which says that macrophage derived extracellular succinate, sort of helps to suppress chronic neuroinflammation, they asked the question whether NSE derived EVs are trafficked into mononucleophagocytes, which are basically cells that will modulate their activation, or their fate basically will determine the level of inflammation that they produce through the signals that they are producing. So to check this, basically the model is to have macrophages grown in vitro. Then you treat them with lipopolysaccharide, which activates them, basically makes them pro-inflammatory. So they give out signals that enhance inflammation. If now to these pro-inflammatory macrophages or activated macrophages, if you give EV-associated EVs, mitoDS red positive EVs from neural stem cells, what happens to these cells? So the effect they have seen through various methods. Again, they're looking at oxygen consumption rates. They're looking at how the EVs are entering into the cells. So let's go through that one by one. So first of all, whether these EVs are entering these macrophages. And so using flow cytometry and plotting the density after flow cytometry, they are able to see when they treat activated macrophages with exosomes and when they treat them with EVs, how the percentage of mitoDS red positive cells are increasing. So easily from this bar graph, you can see that there is a significant increase in the mitoDS red positive cells. And this is further can be seen here in this micrograph, where you see mitoDS red as these tiny red dots. And you see that they are definitely interacting with the activated or LPS treated macrophages, and then these mitochondria are getting incorporated into these target cells. So then if they are actually entering the target cells, what is happening to them after they enter? So what is the intracellular fate of these incorporated mitochondria? So using spin disk microscopy, they generate these very information dense images, and they check whether these mitochondria that are taken up, whether they're going to get degraded by going to the lysosomal or peroxisomal pathways. And what they find is that actually, so here you see mitoDS in red, and LAMP1 is stained in green as a marker for lysosomes, and here PMP70 is a marker for peroxisomes. So when they look at the co-localization of the mitoDS with either LAMP1 or PMP70, what they find is that only close to 7% of the mitochondria are going to the lysosomes or co-localizing with the lysosomes, and about 10% with the peroxisomes. And on the other hand, if they look at the existing mitochondrial network within the activated macrophage, which is seen in green here, and the mitoDS is red, so you can see areas of yellow here, which basically indicate the co-localization of mitoDS, that is exogenous mitochondria that are taken up through EVs. And here in the non-activated macrophages, the percentage of overlap is way less than when we compare it to the activated macrophages. That means that activated macrophages take up more of these EVs. So here, if you see the quantification, about half of them are attached, and about 17 or 18% are included within the host mitochondrial network, which they find out through these imaging data. So the intracellular fate is that they do not get degraded, but get integrated into the existing network. So again, they try another method to check the same, which is basically they use a red fluorescing molecule, which is SF cherry. So they are split molecules. Half or a part of the molecule is already present in the target cell's mitochondria, and the other half is present in the mitochondria in the EVs. So when you look here, this is my macrophages that are activated with LPS, and they have split split fraction of a cherry 2, which is starting from 11. And here in the EVs, you have the same molecule 1 to 10. So a part of it is in the EVs and a part in the endogenous mitochondria. And when they come together, you see fluorescence. So, and this this is used as a control. So basically just lipo, no presence of SF cherry 11 basically. So if you look at the fluorescence then after treatment of EVs to these activated macrophages, there is quite an increase, significant increase in the fluorescence, which means that the exogenous mitochondria are incorporated inside the existing network of mitochondria. And they also do the same, try to find the same thing through confocal, through TEM and confocal microscopy. So here, activated macrophages are again treated with EVs, and they look for mito-DS and nucleus is stained with TAPI. And you see these asterisks on the TEM scanning, sorry, not TEM. These are images, and when you scan them, all these asterisks actually denote the mito-DS red positive foci. And this is basically pseudo color blue given to the nucleus. So here also you see that there is a clear overlap between the mito-DS from the exogenous mitochondria and the activated macrophages. So the majority of EV-associated mitochondria escape the lysosomal peroxisomal pathways, and they co-localize and fuse with pro-inflammatory macrophages endogenous mitochondrial network. So they have these EV-associated mitochondria have found their way into activated macrophages, and they're able to incorporate with the existing network. So since they are getting in there and getting incorporated in the network, what is the way through which they have been taken up by the cells? So what is the method of uptake? To check if it is endocytosis, what has been done is, again, macrophages treated with LPS that is activated, and then after 30 minutes of this activation, inhibitors of various routes of endocytosis are given to these cells, and then EVs and other control particles are given as treatment. And then the staining is done to see whether they have been taken up or not. So basically, if in the presence of inhibitors, these particles are still getting into the cells, it means that there is some other route apart from endocytosis. Cyto is cytochalasin D, which inhibits actin-mediated endocytosis, and DP refers to dinosaur and pitstop, which inhibit the clathrin and dynamin-associated endocytosis. So here you can see that there are three things here. LPS is for activation, and these three are the treatments, particle treatments. So first is the EV preparation, which have mito-DS positive mitochondria. There are latex beads and there are liposomes, which are used as controls here. So let's first look at the controls. The liposomes, you see when the macrophages are activated, you see an increase in the liposome concentration uptake, basically. But when you give either of the inhibitors, dinosaur pitstop or cytochalasin D, the uptake of liposomes drops heavily, which means that the pathway inhibitors are working and liposomes are not taken up. For latex beads, whether if you inhibit the actin-mediated endocytosis, only then the latex bead uptake gets reduced. Otherwise, it's there. But when you come to EVs, this uptake is reduced whether you use any of the two inhibitors. So basically, endocytosis is the pathway through which these EVs are taken up by these activated macrophages, and the same is evident in microscopy as well. So these are control macrophages. basically they're not activated, not given LPS, so the EVs are not taken up. When there is LPS, that there is activation, so you see more of the mito-DS interacting with the membrane and co-localizing there. And when you give them the inhibitor of endocytosis, then you see the situation reverting to control, which means they are inhibiting the uptake of EV-associated mitochondria. Next, the EV-associated mitochondria are taken up by endocytosis, we know, and then they are incorporated inside the target cells. So what are the kind of effects that can happen once they are inside? Upon macrophage activation, one of the things that happens is that the mitochondrial network, mitochondria start to show fission. So basically, these micrographs, the green fluorescence in these micrographs is quantified in this bar graph. So let's look at this. And you see here when LPS is absent, when the macrophages are resting, the fused mitochondria are high in percentage. When you activate them, the percentage of fused mitochondria reduces, the fission is increased. And when you give them the mitochondria containing EVs, this percentage of fused mitochondria increases again, which means these EVs are stimulating mitochondrial fusion. Then intermediate and fragmented mitochondria are also looked at, and you see here that the intermediates basically they are neither completely fused nor completely fragmented nor completely fused, and that is increased when there is activation through LPS, and it reduces when the EVs are given. So this indicates that stimulation with LPS promotes mitochondrial fission, and then EV-associated mitochondria increases fused mitochondria inside the target cells, which are macrophages, activated macrophages here. So they looked at an RNA expression profile using microarray of these target cells once they have taken up EVs. So activated macrophages once they have taken up EVs versus the control, that is those that have not been given any EVs. What are the pathways that are enriched? Through RNA expression analysis, they found that ribosome related pathways, carbon metabolism, and OXPHOS or oxidative phosphorylation are the processes that are upregulated. So since OXPHOS is a direct consequence of having healthy mitochondria or not, they looked at a lot of assays. So extracellular flux assay of oxygen consumption rate, basal oxygen consumption rate, and ECR or extracellular acidification. All of these are big terms, but they're basically showing us the activity of mitochondria inside the target cells. So first of all, the basal oxygen consumption rate when macrophages are given EVs is increased as to when they are not given EVs. Secondly, the basal oxygen consumption rate versus here when you look at the consumption, the oxygen consumption rate here, which is in picomoles per minute here versus time. When there are only resting macrophages, it is at this level. And when they are activated, it drops down. But when you give them EVs, they rise up again. The oxygen consumption rate of activated macrophages is rescued when you give them EVs from neural stem cells. And this clearly indicates the effect of healthy mitochondria that are taken up with the EVs. Lastly, they look at the inflammatory signals. So interleukin, NOS, and IL-6, they are looked at. Their expression is also looked at. So here the interesting part is that when the EVs are coming from FCCP treated neural stem cells, the rescue in OCR as well as the inflammatory signals that are produced by the activated macrophages are increased, which points to the fact that the transfer of EV-associated mitochondria inhibits the metabolic switch of pro-inflammatory mononucleophagocytes. Lastly, they want to see whether these effects that have so far been seen only in vitro cell cultures also occurs in vivo. So they have taken up mouse models, experimental autoimmune encephalomyelitis model. And there is a score given for this particular disorder. Increase in score means there is severity in that disease, and decreased score means there is recovery from that disease. Here, once they have developed this model by EA immunization, then they give injections of NSCs or EVs derived from NSCs, and they look at the neuropathology at day 55, till day 55. So on day 15, they give the injection, and here you can see that PBS injected mice, which is basically a control, they do not show a recovery in the EA score, whereas NSCs and EVs derived from NSCs are able to recover the score, decrease that over a period of 55 days. Lastly, the last set of data shows us that transplantation of NSCs, what happens when you transplant NSCs into this mouse model, and how this disorder can be recovered. And here they have looked at all the various cell types inside the CNS, basically neurons, astrocytes, oligodendrocytes, and monocytes or macrophages. So these are astrocytes, these are neurons, and here you see that you have to look at the mito-DS red spots, which are basically bright red spots, and how they co-localize with the marker for those particular cells. So here it is GFAP for astrocytes. So it has done for this imaging has been done for all the 5 different cell types. Finally, we see that compared to the non-immunized mice or control mice, in EAE mice, when they're treated with NSCs are transplanted into them, the percentage of co-localization is maximum in GFAP positive cells, that is astrocytes, and the macrophages. So this marker is for T cells. These are oligodendrocytes, neurocytes, and macrophages. So transplanted NSCs can transfer the healthy mitochondria to mononuclear phagocytes and astrocytes in vivo. So the effects that we have been seeing so far in vitro culture, they are corroborated by the data that we find in this in vivo in this EAE mouse model. And that concludes the paper. So I think I can open the floor to Q&A. These are some of my questions. In the immunoblots in the beginning, NSC lysates do not show any CD9. I'm sorry, this is a typo. Not CD60, CD9 signal in the first immunoblot that we show. In nano-FCM, I was curious to know what would be the percentage of only the mitotracker positive population as a percentage of the whole. So we talked about more than 3/4 of them being CD63 positive. And second thing from the same data is, why are some EVs showing CD63 negative? So do we have some other particles there which might be of interest? And lastly, what are your guesses about how mitochondria are loaded into EVs? Those are the questions I would want to ask. Thank you.
Thank you, Nishtha, for that wonderful presentation. And so I see that there's a lot of questions in the chat box already that have been answered. I think we can go through Nishtha's questions first. Luca and Stefano, if you could answer any of these questions on the slide right now, and then we can go through the chat box later.
Let me thank the presenter for giving a very good overview of the paper and for highlighting a number of key questions that we don't have an answer yet for. So you did a very good job, and thanks very much for highlighting these critical aspects of our paper. Luca, would you like to try to answer to these questions?
Yeah, so regarding the immunoblots, I think you're referring to CD9, but basically you can clearly see that there's a clear band in EVs. I'm sure that if we would have exposed more the NSC lysates, we would have found also the CD9 in that band, in that lane, but it's clearly much more in the EV preparation as expected. Right. Yeah, regarding the nano-FCM, I think that's very interesting to ask. So essentially, if you look at the figure 4b, the plot is basically divided in 3 major areas. And the ones that are double positive for CD63 and the mitotracker are around 26% of the whole EV population, which is kind of in line with the 10 data that we also obtained. And while some EVs showing as CD63 negative, that's something that has happened in our preparation. I'm not sure how to reply to that. Yeah.
And so I'm just gonna go over some of the questions here in the chat box because this is being recorded, so the questions in the chat box will not appear in the recording. So I'm just gonna read through some of the questions here. So we have the first question we had earlier was actually by Lilia, and she asked about the filtration method, and if you guys think that the filtration was reducing the mitochondrial complex signal from the EV sample. And Luca, you answered that the filtration probably left only mitochondrial fragments in the preparation. And Stefano followed up with, we should have quantified EVs and mito-EV frequencies at the TEM also after filtration. And then Artem asked about, what about apoptotic bodies? Could it be possible that mitochondria's inside apoptotic bodies instead of EVs? And they showed that Western blot showed no evidence of apoptotic bodies in their EV preps. I think someone had a follow-up question with that about which markers you use for apoptotic bodies. Could you please answer that?
Yeah, Camille, we've been using histones in previous papers, and there is no changes in the protocols for collection of EVs ever since we started in the field. Plus, both transmission electron microscopy and cryo-EM failed to show any evidence of apoptotic bodies whatsoever in our mito-EVs preparations.
So Artem, does that answer your question? So if you have any follow-up questions, you could unmute your mic for about this question on apoptotic bodies. Otherwise, we're gonna go to another question. Okay. So we're gonna go a bit over time, at least like 5 more minutes to go through the questions, if that is okay with you guys. If you can stay on for 5 more minutes. So this other question was, is there any evidence that this EV-associated mitochondria transfer pathways occurs in vivo, aside from transplantation in your system?
Yeah, I was about to answer to that. I think it's a great question. So, as I mentioned, there is evidence of in vivo transfer between astrocytes and neurons. So, neural stem cells are like precursors of astrocytes, and possibly they would do the same thing. Of course, in our model, we use purposely a model of inflammation because we wanted to look at the transfer from neural stem cells to inflammatory cells, specifically macrophages. But in the future, it will be quite interesting to understand if this is an homeostatic mechanism by which neural stem cells do communicate with surrounding cells within their niches.
Okay, Roberta, could you please unmute your mic? You have a question here about the LC3 on the mitochondria containing EVs. If you want to unmute your mic, you can ask your question.
Hi, yeah. In our work, we found that LC3 or mitochondrion site autophagosomes can be ejected from the cell as EVs. And I was wondering if you had checked for LC3 or other autophagy or mitophagy markers on your mitochondria containing EVs.
So we haven't, but that's absolutely something very important to assess. So part of the reasons why we went to do this endeavor of looking into neural stem cells is because we came from previous evidence from mesenchymal stem cells, and there are several papers, including one from Finley in 2015, that shows that the mesenchymal stem cells can transfer the mitochondria by these to macrophages to outsource mitophagy. So that's something that we kept in our mind. But being that we found so many active and functional mitochondria, we kind of did not look into that aspect anymore. But that's something that I think definitely needs to be done.
Okay. So there's this other question about from Federica. How do you justify that CD63 are positive for mitochondria proteins? Their dimensions should be much smaller than mitochondria. And Stefano answered that earlier with, we do not yet have anything other than extracellular mito surrounded by an EV-like, at least in terms of proteins or protein markers membrane. Federica, if you have a follow-up question or if that answers your question, I don't see her on the call. So we'll move on to, let me look for the other questions here.
Can I ask?
Yes, you can go ahead and ask.
Per the EAE result of this spectacular piece of work, do you think the entire effect, at least on the EAE, is just due to the mitochondrial transfer? The entire effect of the stem cells?
Thanks, Phil. You're too good to with us for the spectacular. I don't think we can conclude that extracellular mitochondria or mitochondria-associated vesicles or vesicle-associated mitochondria do the whole job. We have showed previously that there is other mechanisms which go beyond the intracellular transfer of mitochondria. So I think it's one of the potential mechanisms.
So Yukai had a question about how many mitochondrial were taken up by each neuron cell on average. And I think Stefano answered this question earlier, that they show the data in the incorporation of macrophages in that paper in the percent of positive cells in figure 6b of the particles or cells. Do you recall what the number is in terms of the percent of the positive cells in the incorporation of these EVs?
Yeah, so I think if I remember correctly, at 6 hours in LPS stimulated macrophages was basically more than 90% of the cells did incorporate these mitochondria from EVs. So it seemed to be very high. As per the number of particles per cell, I think it's shown in figure 7C.
Okay, so in the interest of time, I think that would be the last question. That would be the last question. So thank you, Stefano and Luca, for joining us today, and thank you, Nishtha, for that wonderful presentation again. Thank you for joining us today. Have a good week ahead.
Thank you all for hosting, and great journal club. Thanks very much.
Thank you. Bye now.